Seven Sense Theater. Yeah, today I want to take you through what we call a neuron. A neuron is a cell, the basic unit of the nervous system. So, David7SenseTheater@gmail.com. You can contact us for more and better videos. We have it out our channel, our YouTube channel. Don't forget to like our videos. Yeah, subscribe for more and better ones. And share the videos to other students, share the channel to other students and other people to also enjoy the science we give. All right, let's continue right away. Uh, if you look at this structure, I'm going to talk about first the structure of a typical neuron. A typical neuron or a typical nerve cell is just as you can see this. This is how it looks like. It has the cell body here, which they call, or which we can call the soma. Then it has the longest process, as you can see here. Long process. This one is the axon, and the axon terminates in two botons or axon terminals. You see here. These are responsible for forming the presynaptic knobs. Okay, so the axon is myelinated, as you can see, but there are some points where myelination does not occur, and that is what we call a node of Ranvier. And there are a couple of nodes, and they are very important for increasing the speed of impulse transmission. The velocity of impulse transmission is increased. You shall see this more about when it comes to action potential. But to just give you a hint that where there is a myelin sheath, this one here, this is a lipid, and it is a poor conductor of current, so of electricity. So when the wave of depolarization is setting off from the hillock here, going towards the terminals, this wave of depolarization will not occur at these points where there is myelin sheath, but otherwise it will be leaping from one node of Ranvier to another, to another, to another node. And this will increase the speed of impulse transmission as compared to what would occur if there was a stepwise. If there was from one point to another, point to point transmission, okay? So that is what they call saltatorial conduction. Alright, if I want to talk more about the myelin sheath, it is synthesized by this cell, the Schwann cell, on the peripheral nervous system. And if it was the CNS, the Schwann cell might be represented by the oligodendrogliocytes. So the initial segment is here, and this is the axon hillock. Okay, then this is the nucleus within the cell body. The cell body has dendrites, which are also processes of the nerve. We have the nuclear granules. These we shall talk more about them within this very video. And the nucleus, they are important in protein synthesis. And the nucleus is here. And lastly, I want you to know this. And this structure you can see here, the centrosome is absent. This indicates or is indicative of the fact that the neuron has lost its ability to divide. Okay? Thus, neurons once destroyed are only replaced by neuroglia only. This one can no longer divide. There are no centrosomes. Alright, so let's continue. Let's talk more about the nuclear bodies or granules. The nuclear granules are basophilic granules when seen under the electron microscope. These bodies seem to be composed of rough surface endoplasmic reticulum, and they are present, and the presence of abundant granular endoplasmic reticulum will be indicating the high level of protein synthesis within the neurons. And proteins are needed for maintenance and repair and for production of the neurotransmitter and enzymes. These enzymes are proteins, and the high or abundance of endoplasmic reticulum, the rough one, indicates the increased or higher protein synthesis within the nerves. Enzymes are required to synthesize the protein channels, the sodium channels, synthesize the enzymes which synthesize the neurotransmitters, and a lot. So these nuclear bodies are present in the dendrites as well, but are usually absent from the axon hillock and the axon. Okay, these nuclear bodies disintegrate into fine dust, which finally disappears on fatigue. So on fatigue, they're absent. This is a term which we call chromatolysis. That's how they disappear, the nuclear granules. And we're going to talk now about the types of the neurons. Neurons have different types. As you can see, we have A, which they call unipolar. We have B, which you can call pseudo-unipolar. We have pseudo-unipolar and unipolar. They're a little bit different. Okay? But the point is that we shall talk more about this. Then we have C, which we call bipolar. It has two poles, okay? This one has one pole and pseudo-unipolar has one pole. So this one is having two poles. Now we have multipolar, which has a couple of poles from which the process is arising, okay? Several poles. And this is a classification depending on the number of poles. That's how we call it unipolar, pseudo-unipolar, bipolar, and multipolar. But now, if I want to talk more about the unipolar, these are true unipolar cells, and they are present only in the embryonic stage of human being. However, the primary sensory neurons, or the neurons conveying impulses from a sensory receptor to the spinal cord, are pseudo-unipolar. So the primary sensory neurons are pseudo-unipolar. This is very true. Okay, so signals carrying pain, signals carrying taste, signals carrying light. Okay, they are all pseudo-unipolar cells carrying this information. Did I say light? Olfaction. Okay, they are unipolar cells carrying signals of senses. Okay, now we have bipolar neurons. Bipolar neurons are found in the vestibular and cochlear ganglia and in the nasal-olfactory epithelium, as well as bipolar cells in the retina. The multipolar cells, most vertebrate neurons are multipolar, especially in the central nervous system. The dendrites branch profusely to form the dendritic tree. So the dendritic tree comes in at around the multipolar system, and you'll see that question coming to you soon. Okay, depending upon the function, that is the second classification of the neurons. And depending upon function, there are two: motor and sensory. If I just separate or isolate out two neurons, and one is motor and one is sensory, how can you classify that this is a motor neuron? Look at this. Motor neuron or efferent nerve fibers carry the motor impulses from the central nervous system to the peripheral effector organs like muscles, glands, and blood vessels. These neurons have very long axon and short dendrites. You see this? Very long axon, short dendrites. Then you know that is a motor neuron. When it comes to sensory neurons, these are afferents, and they carry the sensory impulses from periphery to the central nervous system. You'll find they have a short axon and long dendrites. So that is a sensory. Short axon, sensory. Long axon, motor. Okay, the opposite of the zones of the neuron. If you find a nerve cell here, it has several zones. One has the receptor zone. Here, as you can see, I labeled. This is a region for graded electrogenesis. Okay, this is dendrites or receptor zone, and has the site for origin of the conducted impulse, because when it originates here, the impulse will be conducted through the longest process, that is the axon. And the site of origin of conducted impulses is here, and it can be the initial segment and axon hillock, as we saw in the labeling of that parts of the neuron. Then the axon forms the zone of all or non-transmission. At this point, you either transmit or do not. Okay, all or non-transmission. Then the last part at terminals, this terminals are zone of secretion of the synaptic transmitter, and this form the nerve endings. Okay, so if I can just go through them, the receptor zone or dendritic zone is where the local potential changes are generated by integration of synaptic connections. You can find that a couple of presynaptic knobs are converging, and they're sending several neurotransmitter which will generate local potentials within the dendritic zone. Now, the site of origin of conducted impulse is the site where propagated action potentials are generated. In the case of spinal motor neuron, initial segment, and in cutaneous sensory neurons, the first node of ganglia. Are you getting my point? That the site of origin of conducted impulse for a spinal motor neuron will be the initial segment, but for a cutaneous sensory neuron, it will be the first node of ganglia. Alright. Zone of all online transmission is what we call the axon. And lastly, the zone of secretion of transmitter. These are nerve endings. The propagated impulses, call them action potential to the nerve endings, cause the release of the neurotransmitter. That's why the neurotransmitter is released, just because of the arrival of the impulse at the axon terminals. So now we've gone to another step of neuroglia. We've said about the neuroglia. Uh, These are glial cells. You can just call them glial cells. And they are just supporting cells present within the brain and the spinal cord. That is the central nervous system. They are numerous and about 10 times more than the neurons. Glial cells may be divided into two major categories. You're going to see those categories, and they will be microglia and macroglia. But if you just look at these diagrams here, you have different types of glial cells, and the A are the fibrous astrocytes. Okay, this is a fibrous astrocyte. Fibrous astrocyte. Astrocytes are divided into two more groups, so this is the fibrous form. Then the B is the protoplasmic astrocyte. Protoplasmic astrocyte. Then C is the oligodendrogliocyte. This one. And the D is the microglial cell. Okay, so this is a microglial cell. If I have the neuron here, the supporting cell here is the microglial cell. You can see it here. All right, let's continue. And the types of neuroglia, I'm still continuing with them. One, we have macroglia. Macroglia are ectodermal in origin, as opposed to the microglia, which are mesodermal in origin. So the macroglia are ectodermal in origin, and these are of two types. You have astrocytes and oligodendrogliocytes. So the astrocytes may be further divided into protoplasmic or fibrous astrocytes. Okay, and the microglia, or what you can call the small glial cells, are mesodermal in origin. These are the smallest neuroglial cells, having flattened cell body and short processes. They are more numerous in gray matter than in white matter. These act as phagocytes and become active after damage to the nervous system by trauma or by disease. That is the function of microglia. They are phagocytes. Okay, we've seen one attached towards the soma. Then the oligodendroglyocytes will be for producing the myelin sheath in the central nervous system. Then the astrocytes are supporting cells. Let's continue. Now let's look at the peripheral nerve. The peripheral nerve, as you can see here, okay, a peripheral nerve is a special nerve. When you have a group of neurons, when you join them together, you'll form what we call a nerve, okay? Because a nerve is a nerve has several cells. Those cells are the ones we've been calling neurons. So when you have a peripheral nerve here, if I just take a look at this cross section, you'll see that I have a lot of nerve fibers being enclosed in a fasciculus or fascicle, as you can see here. And these bundles or fascicles are the ones which are forming what we call a nerve. Okay, so you can see seven neurons forming just a single nerve. And if I just take you through, the outermost part is the epineurium of the nerve. Okay, and if I go inside, you'll find that if I just focus on one fascicle, this outer part is called the fasciculus. This entire thing is the fasciculus, and its outer part forms the perineurium. Okay, this is epineurium, this is perineurium, and the perineurium just encloses several neurons. And if you try to go a little deeper, now you have entered to a single nerve fiber, and that outer part forms the endoneurium. Okay, and the endoneurium, you can see here we have the inner lamina of the perineurium. Okay, this is a perineurium will be having two lamina. So this inner lamina of the perineurium, and the inner lamina of the Schwann cell is also visible there. If you can just see, you can see something there. All right, so we can continue right now. This is just an axon. Okay, so all right, let's continue about the peripheral nerve. A compact band of axons located outside the CNS is called a nerve, a peripheral nerve. And in this nerve, the axons are arranged in different bundles, which we call the fasciculi. Each axon or nerve fiber is covered by endoneurium, which is bounded internally by basal lamina around the Schwann cell and externally by relatively impermeable inner basal lamina of the perineurium. Okay, we've seen this. And each fasciculus is covered by perineurium. The cells of the perineurium are tightly adherent and act as barrier to the passage of particulate traces, molecules or toxins into the endoneurium. So the whole nerve is covered by epineurium, which limits the extent to which the nerve can be stretched by body movements or external pressure, thereby protecting the fragile nerves or fragile axons, fragile neurons or fragile axons inside the nerve. So the nerve, you can see an axon is highly protected by a couple of layers, call them endoneurium, perineurium, and we've seen the epineurium. Okay, so don't you forget to like our videos or subscribe for more and better videos, and share the videos to other students to also enjoy. The video 7 Science Theater. Thank you.